We will email the recording of this event to those of you who have registered for the event. This recording will be up for probably about 2 weeks after today, and then at which point it will be taken down. So just keep that in mind. Um, if we have any time at the end of today's event, we may be a little tight, but if we have any time for Q&A, that'll be held towards the very end. So at that point, um, we'll ask for questions in the chat if we get time. I'm going to thank AbbVie, our sponsor for today's event. Without them, we wouldn't be able to host events like this. And then our topic for today's event is a really exciting one. This is the history of chronic lymphocytic leukemia with Dr. Jeff Sharman. So Dr. Sharman is our speaker for today. He is the Director of Research at the Willamette Valley Cancer Institute, as well as the Medical Director of Hematology Research for the U.S. Oncology Network. So we are honored to have you here, Dr. Sharman. And with that, I'll turn it over to you.
Great. I'm going to go ahead and share my screen here, I hope. Share. And let me put this over here. Hopefully my slides. Are you seeing my slides currently?
We are seeing the Zoom home screen thing.
Okay, let's try this. Oh, there we go. All right.
There you go.
Got it. Okay, well, the title of my talk today is CLL Mission Accomplished, 100 Years of Accelerating Progress. In May 1st, 2003, President Bush declared mission accomplished from the deck of the USS Abraham Lincoln, 6 weeks after the start of hostilities in Iraq, declaring major combat operations in Iraq have ended. But the war would last another 10 years with unacceptable loss of life and resources. In many ways, we're in the same place with CLL. With the introduction of novel agents, we've seen survival curves very close to age-matched controls, and chemotherapy has been banished. Yet for our patients, let's see, where we go. Yet for our patients, therapy remains a constant source of anxiety, and cures remain elusive. In the next hour, I would like to walk you through the history of CLL, how we've arrived where we are today, and I'd like to introduce you to the names and ideas that have propelled the advances. While some of the stories may be familiar, I suspect many of them will be new and surprising. There's a lot to cover. I have 120 slides and 60 minutes, so let's jump right in. Leukemia was not even recognized as a disease until the 1800s. In 1811, the first accepted clinical description of leukemia occurred when Peter Cullen described a case of splenitis acutis, in which the serum of blood had the appearance of milk. He had examined a 35-year-old male patient who presented with pronounced abdominal pain and symptomatic fever, and the patient was treated with bloodletting for 5 times, but died shortly thereafter. But leukemia was a disease for which the microscope would be essential. And despite their invention in the 1500s and 1600s, they wouldn't be applied to medical research until the early 1800s. Medical microscopy was popularized by Alfred Donnet at La Société de Médecine in Paris in the 1830s to the mid-1850s. Donnet was a pioneer in microscopy for the study of diseases, yet many questioned whether the microscopic elements of life had any bearing on health. In 1939, a 44-year-old woman with splenomegaly died under the care of Dr. Barth at the hospital where Donnet worked. Donnet examined the blood of the patient and observed that half of the corpuscles, this is a quote, half of the corpuscles in the blood appeared like mucus globules. That's shown here in the middle. Donnet briefly described this observation in a manuscript entitled Cours de Microscopie in 1844. And if you examine the unstained slide, there are a few distinguishing features of white blood cells. Initially, they were thought to reflect mucus in the blood. Virchow in 1847 was the first to recognize what we now call chronic lymphocytic leukemia and distinguish it from the myeloid version of the disease. He had already described 9 cases of leukemia associated with gross enlargement of lymph nodes and spleen. His first patient had developed gradual, painless enlargement of the cervical, axillary, and inguinal nodes over a period of 2 years. And after his death, the spleen, though firm, was not enlarged, and Virchow later associated this type of disease with the small lymphocyte cells in the blood, similar to those present in the lymph node. But it was Paul Ehrlich who added color at the turn of the 20th century. Ehrlich was a remarkable individual who could reasonably be credited among the fathers of hematology, immunology, and even antibiotic therapy. He pioneered the use of animal models to create a drug called salvarsan for the treatment of syphilis. And in the process, he actually invented the term chemotherapy, which was a contraction for chemical therapy. That term would be applied to subsequent generations of antibiotics. With training in dye making, he learned that the different pieces of the cell could be stained, transforming the mere mucus globules into a vibrant world of white blood cells. And this would empower the field of hematology by displaying the rich diversity of blood cells. From 1900 to the 1940s, recognition of the disease and early attempts at the management were explored. With greater ability to distinguish among white blood cells, it was George Minot who described a series of 87 cases of lymphatic leukemia in 1924. Although describing the cases as chronic, several were diagnosed before the age of 20 and even a few in infancy, likely other forms of lymphoid malignancy. Radiation had already been established as a mean to treat enlarged lymph nodes, and he would subsequently receive the 1934 Nobel Prize in Medicine Physiology for his work on pernicious anemia. 20 years later, only modest changes had been made. In 1942, the state-of-the-art remained radiation, but the ability to distinguish between the various variations of lymphoid malignancies had improved. Here, you can see that they distinguished among lymphoblasts or lymphosarcoma cells and CLL representative different diseases. Describing therapy, they wrote, quote, a series of daily treatments, numbering usually 8 to 12, is applied regionally to the peripheral lymph node areas, the mediastinum and the retroperitoneal region, and the spleen. The indications for therapy are anemia, debility, evidence of increased metabolism, pressure symptoms, and least important, a progressive rise in leukocyte count. Aside from external beam radiation therapy, systemic therapy was primitive. In Portland, Oregon, Edward Osgood developed the use of radioactive P32. Reflecting on his own experience, here he writes, quote, a few patients were observed for a period of months to rule out infectious lymphocytosis before therapy was started. But in the majority, therapy was started the same day they were first seen. The patients who had mild symptoms felt so much better soon after the start of therapy that it was decided to begin immediate therapy for all patients, including those who claimed to feel perfectly well. In England, it was the pioneering work of Alexander Haddow, an individual who returns to our story later, who was the first to demonstrate the use of pharmacologic therapy for CLL. He published his work in The Lancet showing the effects of urethane. Here he writes, quote, after experimental work on the growth of inhibiting effects of urethane on animal tumors, it was decided in 1943 to test the drug in advanced cases of malignant disease in man. In some of these, a fall in the leukocyte count was noted, and this suggested the trial of urethane in patients with leukemia. For those familiar with hematology, you can note the chemical similarity between urethane and hydroxyurea, a drug we still use. World War II, however, ushered in an enormous number of scientific advances, and it was the watershed period in our understanding of cancer. If World War II was, quote, the Big Bang, end quote, of modern science, it was Vannevar Bush, with no relationship to George, who spoke it into existence. An engineer from MIT moving in Washington, D.C. circles, he recognized that they were woefully underprepared for war. Gaining an audience with Franklin Delano Roosevelt after the fall of Dunkirk, a 10-minute meeting would lead to the creation of the Office of Scientific Research and Development, known as the OSRD, which would come to employ 30,000 individuals and 6,000 scientists to organize the scientific community in a program of what was known as industrial science. Notable achievements of the OSRD included the Manhattan Project, development of radar, and synthetic rubber to replace the natural supplies when the rubber trees of the South Pacific were lost to the Japanese forces. Within the OSRD, however, was the Committee for Medical Research. 4 revolutionary treatments came from this group. Although penicillin had been discovered a decade earlier, it couldn't be synthesized in sufficient quantities until scientists working at a small New York chemical company that had never before produced a drug figured out how to ferment it in collaboration with the OSRD. That would become Pfizer. In the Battle of the Pacific, a number of soldiers who died from malaria was about the same number lost to combat. A large screening program with testing conducted in conscientious objectors and prisoners seeking to obtain earlier parole led to the discovery of chloroquine. With propaganda images supplied by Dr. Seuss, the screening program would serve as a model for the subsequent efforts to discover chemotherapy. Furthermore, prominent scientists from the program, including Gordon Zubrod, would go on to lead the National Cancer Institute. The Blitzkrieg was fueled by methamphetamines. German soldiers were given Preventin, which was a drug they used, allowing them to stay awake for days at a time. The absence of sleep allowed them to move forces at rates never before anticipated, leading to the immediate collapse of French and British forces. Synthetic steroids did not exist, but adrenal cortical extracts were thought to enable pilots to fly at greater altitudes. So the Office of Scientific Research and Development poured resources into the development of synthetic chemistry. Rumors of a captured German U-boat transporting bovine adrenal glands from Argentina to Germany triggered panic that enemy forces would soon be able to fly at altitudes unreached by Allied pilots. Steroids will return to our story shortly. But it was a history of mustard gas that serves as an auspicious foundation of modern chemotherapy. Known as the king of battle gases for their impact on World War I battlefield, there was a concerted effort within the Committee of Medical Research to develop medical therapies, clothing barriers, and so forth to protect soldiers. Many are likely aware that early chemotherapy experiments that arose from mustard gas research were conducted at Yale University. Few know why they happened there. The World War II mustard gas efforts were led by Milton Winternitz, a Hopkins-trained pathologist who was the dean of Yale Medical School. As a younger pathologist, he had summarized the medical knowledge of sulfur mustard gas developed in the First World War and published his findings in a 1920 compendia. He was a profoundly controversial character known for his explosive temper. For instance, he was recorded to have thrown phones at his secretary and ended up being forced from the dean position due to his bad behavior. As we'll see shortly, his intellectual dishonesty and idea theft laid the foundation for pioneering chemotherapy research to occur at Yale. In his 1920 publication, Winternitz had neglected to include findings from field medic Dr. Krumbar from the First World War, who had noted that soldiers who had survived the initial skin wounds of mustard gas would develop low blood counts. It was a finding that would have to wait another 20 years for its importance to be realized. The way most people are familiar with the story of mustard gas being discovered to have an impact on blood is the story of Stuart Alexander, a chemical warfare specialist who arrived on the scene of America's second Pearl Harbor at the port of Bari, Italy, as the Allies were advancing north on the Italian peninsula. A merchant ship carrying nitrogen mustard munitions to the front lines was bombed, exploded, and sank. Sailors thrown overboard were coated in nitrogen mustard, but due to the secrecy surrounding the presence of war gases, medics were unaware. It was only the heroic deductions of Dr. Alexander who identified the presence of mustard. He went on to describe the falling white blood cell count among survivors and wrote an exhaustive report from which the link between mustard and lymphotoxicity was described. But the observations of Stuart Alexander were not by chance and circumstance alone. Prior to his role as a field medical officer, Stuart Alexander had actually done secret work at the Edgewood Arsenal in Maryland. This facility had been established by Milton Winternitz for the Committee on Medical Research. Laboratories were established to evaluate the impact of war gases on soldiers and develop mitigation measures. Sulfur mustards had been used extensively during the First World War, but a small sample of nitrogen mustard had been secretly exfiltrated from Germany, and Stuart Alexander had been tasked with studying the effects of the novel chemical warfare agent on laboratory animals. It was here that he found nitrogen mustard effects on multiple animal species and would lead to the depletion of lymphatic tissues. The secret testing conducted at Edgewood would shock anyone. Not only were there animal experiments done here, but throughout the Second World War, fully 60,000 American soldiers were purposefully exposed to poison gases to help determine the effect of the gases on soldiers, develop protective clothing, and medical interventions to protect soldiers should they encounter war gases on the battlefield. These human experiments were conducted under the direction of Dr. Cornelius Rhodes, who would ultimately become an intellectual father of chemotherapy and serve as the initial leader for the Sloan Kettering Institute. But his history of disregard for human subjects would ultimately destroy his legacy, barely receiving a mention on the current Memorial Sloan Kettering webpage. Earlier in his career, he had been conducting human experiments in Puerto Rico for the Rockefeller Institute to better understand pernicious anemia and sprue. His work ended abruptly when it was discovered he had written a highly racist letter, shown here, where he admitted to trying to kill patients by injecting them with cancer. The letter was never mailed, and he alleged it was written sarcastically. And ultimately, with heavy PR lobbying from the Rockefeller Institute, he was cleared in an investigation that some felt was a whitewash of his record. His work on mustard gas, for which the military would be considered appalling by modern standards, would actually rehabilitate his career, and he would rise to become a hero of early oncology. Ultimately, the letter would resurface years later, and his reputation would be forever tarnished. After discovering the lymphocytal activity of mustard in laboratory animals, Alexander saw the potential for treatment of lymphatic cancers. Unfortunately, he was not allowed to pursue this idea further, as he was told by his superiors that the idea was beyond the scope of battlefield readiness research. He was given the opportunity to present his idea, however, to Milton Winternitz, who was still dean of Yale Medical School. In March 1942, his report was presented to Winternitz, who mocked his findings and charged him with sloppy research. Unbeknownst to Alexander, however, Winternitz would immediately secure an OSRD grant to continue that research at Yale University. Frustrated by his circumstances, Alexander would leave the laboratory and become a field medical officer in the North African campaign. That research was assigned to two Yale faculty, Goodman and Gilman, who started their work in early 1942. A single mouse with a large lymphoma tumor was treated with nitrogen compounds and quickly resolved the tumor. Further mouse experiments would follow, but the results of their first remained their best. They persuaded a surgeon to treat a patient with advanced lymphoma whose disease no longer responded to radiation and whose condition was described as, quote, utterly hopeless, end quote. Due to the secrecy accompanying the use of classified war gases, it was merely described as lymphocytal substance X. With a tracheostomy kit at the bedside due to the impending airway obstruction, J.D., a Polish immigrant, was treated in December 1942. With no IRB oversight, no phase 1 dose finding, no informed consent, or any of the modern elements of clinical research, he was injected with an IV version of nitrogen mustard. Here he is shown before on the left and after on the right. Prevented from pursuing what he considered to be valuable research and unaware of the work that had started at Yale University due to secrecy considerations, Stuart Alexander moved to the battlefield of North Africa, where he became a confidant of General Eisenhower. Flown to Italy immediately after the bombing of Bari, Italy, he became convinced that nitrogen munitions had been present. He recognized that he was given a once-in-a-lifetime chance to document the impact of nitrogen mustard on human blood cells and carefully documented the impact on lymphatic tissues and marrow. His controversial report was rejected by Winston Churchill, who refused to acknowledge the presence of mustard gas in the harbor and led to a tense standoff. FDR, however, quietly acknowledged the accuracy of Alexander's findings. His report was highly confidential, but one copy was quietly requested by Milton Winternitz at Yale, who had clearance to review the findings. Following the discovery at Yale University, a small consortium of luminary investigators would explore the efficacy of nitrogen mustard. Due to secrecy, however, it would not be published until after the war in 1946. Quote, encouraging clinical results, sometimes dramatic, have been obtained, particularly in Hodgkin's disease and lymphosarcoma, and occasionally in chronic leukemia. Mustargin would become the first chemotherapy. The drug, however, was quite unwieldy to use clinically. Here they write, quote, local extravasation during the injection of solutions resulted immediately in pain and subsequently in tender indurated swelling and resolved slowly. Remarkably, this drug was still in use during my fellowship for the treatment of Hodgkin's lymphoma. Shortly thereafter, in 1948, Sidney Farber reported the discovery of aminopterin, known as methotrexate. Suddenly, there was a real hope that that cancer could be conquered in the same way that penicillin had transformed infections. The manufacturing capacity and industrial research conducted during the war under the OSRD had propelled the Allied forces to victory. Alfred P. Sloan, the CEO of General Motors, and the chief scientific officer, Charles Kettering, had led the development of countless technologies for GM and were celebrated for being the centerpiece of the, quote, arsenal of democracy, end quote. But they were facing a potential PR crisis. Sloan had actually miscalculated early in the war, estimating that Germany might prevail quickly and wanting to be on the right side of a post-war Europe. Their German subsidiary Opel had provided the trucks and airplanes to German forces. GM had in fact been supplying both sides. To shape the post-war dialogue and capture the early enthusiasm of chemotherapy, they founded the Sloan Kettering Institute adjacent to Memorial Hospital in New York City. In 1948, Cornelius Rhodes, who we met previously, became the director of Memorial after declining an opportunity to work with Dr. George Minot in Boston, the year after he had received his Nobel Prize. He would bring his team from the chemical warfare program, including David Karnofsky, who would lead the clinical aspects of the program. They would be tabbed with launching an ambitious cancer drug screening program that mirrored the malaria research program from the war. Stuart Alexander was actually offered the senior clinician role, but he demurred. Compounds would be screened against animal models and taken to clinical trials if there were any signals of efficacy. At the time, little was known about the biology of cancer, and this was essentially a brute force effort. Among the most commonly used models was an aggressive lymphoma, so naturally many of the drugs taken forward would skew towards activity in hematologic malignancies. Recognizing the limitations of IV mustard gas, they identified a compound called triethylene melamine, which could be given both orally and IV. To provide objective measurements of clinical improvement, Karnofsky developed his, quote, performance status, or the KPS measurement, which is still in use today. Kettering had personal connections to cancer as he had lost his sister to the disease. He provided the key funding to an organization known as the Southern Research Institute, which had recruited Howard Skipper, also from the Chemical Warfare Service, to lead cancer discovery efforts. Skipper would collaborate with Gertrude Elion, the Nobel Prize winning scientist whose career started out testing the pH of pickles and the color of mayonnaise for a grocery chain. The war ended. The war enabled her to join George Hitchings at Burroughs Wellcome, where she developed 6-mercaptopurine that would be tested at Sloan Kettering and found to have activity in acute leukemia. She would receive her honorary PhD only after winning the Nobel Prize. Howard Skipper would become the scientific genius credited with arguing in favor of combination chemotherapy. For his part, Cornelius Rhodes would be known as a pioneer whose contributions elevated him to the cover of Time magazine in 1949. He would become the face of cancer discovery on a national level, but eventually his controversial legacy would emerge. The award given annually in his name at the American Association of Cancer Research would be renamed, and he would die suddenly of a heart attack at work 10 years later. On the Memorial Sloan Kettering webpage, you can read the following, quote, SKI's first director, a pathologist named Cornelius Rhodes, had served as the chief medical officer of the Army's Chemical Warfare Service. His ties to the military and charges of unethical human experimentation would later sully his reputation as a pioneer of chemotherapy. The formation of the National Cancer Institute followed from efforts at Sloan Kettering. Nudged on by the wealthy socialite Mary Lasker of the Lasker Prize and Sidney Farber, Congress allocated funds to expand the chemotherapy screening program established at Sloan Kettering. Partnering with industry, notable drugs to emerge would be vincristine, which was being evaluated as an anti-diabetic agent at Lilly, based upon the folk use of the Madagascar periwinkle in Jamaica, and anthracyclines would be identified to come from the soil bacteria isolated at an Italian castle. Under the leadership of Gordon Zubrod, who had led the World War II malaria program, the solid tumor unit focused on the use of surgery, radiation, and the sequential use of monotherapy. Meanwhile, the hematology branch began an aggressive work in pediatric acute leukemia. It was the laboratory work of Howard Skipper and the clinical efforts of Emil Frey, who would go by the name Tom, and Emil Freireich, his colleague, that would lead to combination therapy regimens. Their controversial development of the VAMP regimen would lead to the very first cures of acute leukemia, management of febrile neutropenia, and platelet transfusions. But their work was so revolutionary that residents refused to work with them, and they were routinely shouted down in meetings. But acute leukemia wasn't their only target. Building upon the initial discovery that adrenocorticotropic hormone from the pituitary could shrink lymphoma, they pioneered the use of steroids in chronic lymphocytic leukemia. Remarkably, they showed that this therapy would cause spleens and lymph nodes to shrink at the same time as white blood cell counts increased, much like modern B-cell receptor signal antagonists. Recent findings published just last year from Lou Stout's laboratory have identified that steroids suppress B-cell receptor signaling, likely accounting for the phenomena of lymphocyte redistribution seen by Frey decades earlier. This is a phenomena common to B-cell receptor signaling inhibitors, but was virtually lost to history for almost 50 years. As brute force compound screening continued at the National Cancer Institute, however, a different approach was taken in England. At the Chester Beatty Research Institute in London, Alexander Hadow, who we had met from his earlier work on urethane, began dissecting how alkylating agents worked. He set out to determine how the drugs worked by modifying the molecule to determine what was essential for it to work and what could be gotten rid of. He hypothesized that some cross-leaking of vital cellular component must be the mechanism of action, and the spacing was really important. He therefore wanted to improve upon nitrogen mustard, and he added an amino acid called phenylalanine to the molecule, and a drug called melphalan was born. He added a slightly different molecule to nitrogen mustard, and chlorambucil was born. And this allowed the oral administration of nitrogen mustard with acceptable toxicity. It's not advancing here. There we go. On the clinical side, Dr. David Galton pioneered the use of chlorambucil in lymphoma and CLL. In particular, his first article was published in 1955. Galton would be the first clinician to really dive deeply into the clinical management of CLL and reported his results of 88 patients managed over 15 years. By carefully plotting the rates of their blood count changes, he anticipated the future discovery of IGHV mutation status several decades before they would be discovered. His review article in 1966 brought the medical community attention to a disease that had otherwise been neglected. Here he presents an algorithm to distinguish between chronic lymphocytic leukemia and small lymphocytic lymphoma when presenting with an enlarged lymph node. William Damashek would be among the first to ever use nitrogen mustard chemotherapy, but he had really become a chemotherapy nihilist. He had also founded the journal Blood, the main trade journal in our field. He stridently objected to the work being done at National Institutes of Health and would write the 1967 review that would serve as really the foundational basis for understanding of the disease for the next 3 decades. Interestingly, nowhere in his review did he mention any sort of therapy other than steroids. He organized stages based upon the progressive nature of the disease, describing them in chronological evolution from asymptomatic stage 1 to an enlargement of lymphatic tissue with occasional constitutional symptoms in stage 2, to frequent symptoms with common infections in stage 3, and to stage 4 disease with organ disturbances in the nasal sinuses, lungs, skin, and so forth, and various other cancers and autoimmune problems with anemia before the patient would experience cachexia and die. There was really no consensus, however, on why some patients with CLL did poorly and others would remain free of symptoms for many years. It was Conti Rai who finally unlocked the mysterious disease. His mentor had tasked him to, quote, figure it out, which is what he set out to do. Not allowed to remove the charts from the medical records department, he transcribed them by hand and organized them by taping them to walls and moving them from wall to wall until a pattern emerged. People called him the, quote, crazy Indian, end quote, who spent weeks organizing and reorganizing charts until he came up with Rai staging. He initially struggled to get his paper published, and it sat on his desk for several months before his mentor pushed him to get it finished. On the right is the first Kaplan-Meier curve we'll see, and these are incredibly important in oncology. The vertical axis measures overall survival, and the horizontal axis measures time. As time progresses, patients die, and you can look at the outcome of different groups. More modern Kaplan-Meier curves might look at progression-free survival or the time you're alive without progression. And if you note in this mid-1970s paper, the median survival was only 2 to 4 years from diagnosis for those with advanced disease and 6 years for the whole group. The following year, the Europeans invited Rai and several colleagues to attend the inaugural meeting of the IWCLL to refine his staging model. This is a meeting that continues every other year and is really a centerpiece for the management of patients with chronic lymphocytic leukemia. But what would emerge from that meeting would be the Binet staging that's really used throughout Europe today. It was planned to harmonize the European and American staging systems, but that never really happened. But by establishing criteria for staging the disease and the precedent for experimentation, the first randomized studies were launched in England under the Medical Research Council. CLL1 combined chlorambucil versus CVP, and no difference was seen, but chlorambucil was much easier, so it won. The CLL2 study compared chlorambucil prednisone versus chlorambucil. Again, chlorambucil was declared the winner based upon simplicity. And if you note that the survival of patients enrolling in the treatment study was really only about 3 to 4 years. Within each of these studies, though, patients with early stage disease were also randomized. And if anything, delayed therapy appeared to be better than upfront therapy. Several studies would be combined, and this would be deemed notable enough to be published in the New England Journal of Medicine in 1998, ending debate about early versus delayed therapy. So chlorambucil with or without prednisone would remain the main tool for CLL management for nearly 40 years until we get to fludarabine. Once again, the story returns to the Southern Research Institute. Chemists there working under Howard Skipper had synthesized several alkylating agents. Building upon the work of Gertrude Elion, they developed fludarabine. As a CRO, Southern Research Institute evaluated fludarabine in several preclinical models and worked out the pharmacokinetics, but a critical error was made that would nearly doom the drug. They developed their pharmacokinetic model in dogs, but didn't realize that there was a profound difference in drug metabolism between dogs and humans. Moving to humans, they miscalculated the suitable dose levels, really resulting in horrific neurotoxicity. You can see this initial dosing in this review and the consequences of severe overdoses in humans. Although it was notably effective in hematologic malignancies, the starting dose had to be reduced by nearly 99% in initial patients who had suffered catastrophic neurotoxicity. One of my colleagues, Dan von Hoff, was credited with observing the profound low white blood cells when the drug was initially evaluated among solid tumor patients. After Tom Fry and Emil Freireich had been recruited from the National Cancer Institute to the newly established MD Anderson Cancer Center, the leukemia program flourished. An enterprising young clinician named Michael Keating undertook the first study of fludarabine exclusively in chronic lymphocytic leukemia among patients refractory to chlorambucil and demonstrated that about one-third of patients could achieve impressive responses that were really quite durable. Among those whose disease failed to respond to therapy, survival was measured in weeks. Those with partial responses only survived about a year, and this would be published in the journal Blood in 1989. Keating attempted to describe response rates, but there was really no language to do so. Bruce Chesson had returned to DC to coordinate what was called the extramural division of the National Cancer Institute in blood cancers. Bruce had been told that he was not publishing enough to make faculty, and his tenure in Salt Lake City was cut short one day when the luminary Max Wintrobe walked into his office and declared it his own. Recognizing that few of the hematologic malignancies had any standardization, he convened groups to standardize disease criteria in multiple disease states. It was at one such meeting, convened in a trailer, where they debated what threshold should distinguish chronic lymphocytic leukemia from small lymphocytic lymphoma. Bruce offered 5,000 as a suitable number, and nobody really debated or contested the number, and so it stuck. Countless patient encounters since have then described the difference between CLL and SLL merely as a number. But emboldened by staging, response criteria, and a novel therapy, a study led by Conti Ryan, the cooperative groups to compare fludarabine to chlorambucil or the combination. Fludarabine and chlorambucil was deemed too toxic and enrollment was suspended, but this was published in the New England Journal of Medicine in 2000, establishing fludarabine as the preferred agent. In 1996, Michael Halleck established the German CLL study group, which would conduct many of the standard-setting studies in the disease. The CLL-1 study compared fludarabine versus watch and wait in early-stage disease, further solidifying watch and wait as the standard for asymptomatic patients. Ian Flynn, a colleague we work with closely, would subsequently publish the ECOG 2997 study, which compared fludarabine and cyclophosphamide versus fludarabine alone in the JCO in 2007. Similar findings were reported by the German group. The first time, CLL began its successful path towards combination therapy, which had been validated in numerous diseases dating back to the original VAMP studies at the National Cancer Institute. But immunotherapy was about to upend convention. As a medical student at Stanford University, Ron Levy attended a lecture given by the iconic Stan Schreier discussing a CLL patient. Ron recognized that the clonality of lymphoid cancers offered a therapeutic opportunity. Cancerous B cells could be distinguished from the benign B cells by their unique B cell receptor. After completing his medical training at Harvard University, he returned to Stanford with an idea. Cancer had never been treated using immunotherapy, and he would be the first to do so. He set out to create antibodies against the unique B cell receptor of each and every patient. There was a new technology called hybridoma technology that was essentially new at that time, and Ron began to produce antibodies against each and every patient's individual B cell receptor while ignoring the benign B cells. Ron's medical student at the time, David Maloney, would treat the first patient and had an incredible response to therapy, igniting hope that such technology could revolutionize therapy. Together with an entrepreneurial fellow named Richard Miller and the financial backing of a Silicon Valley rocket scientist, they founded Biotherapy Systems, a company whose purpose would be to manufacture unique therapy for each patient with lymphoma and leukemia. Meanwhile, in Boston, Lee Nadler identified several B lineage surface markers, including what we now know as CD20, and developed monoclonal antibodies against the target. But making the jump to exploit that therapeutically would occur back west. Biotherapy Systems was going bankrupt and needed a larger partner, so they merged with the San Diego company IDEC Pharmaceuticals. Recognizing the impossibility of scaling the manufacturing of individual patient products, they began vaccinating mice with lymphoma and created unique hybridomas. They settled on one antibody against a B cell antigen and subsequently recognized that it bound CD20. Remarkably, David Maloney had returned for his fellowship and was once again working with Ron. This 1994 paper in the Journal of Clinical Oncology described their first in human phase 1 anti CD20 antibody. In their limited phase 1 experience, they never identified what was called a maximum tolerated dose, and they could not observe improved efficacy with higher dosing. Based upon the supply of the drug and the next study plan, they picked 375 milligrams per meter squared as the halfway point between the two highest dosages. This scan is one of the first patients treated with rituximab and shows a progressive disappearance of a lymph node at the arrowhead there. And this was the first successful use of therapeutic antibody to treat cancer. As 1994 progressed, however, a chance encounter would change the course of lymphoid malignancy research. Genentech executives were visiting Stanford to learn of new scientific discoveries. Ron entered the lunch hall, and as an introvert, he sat at an open table. He was subsequently joined by David Ebersman, in charge of business development at Genentech. Neither knew each other, but as they began talking, they realized their interests aligned. IDEC was running out of money, and Ron needed a larger partner. And based upon that random lunch meeting, Genentech would acquire rituximab and help set the direction of Genentech for the next 2 decades. At this point, though, the leading institutions in CLL research had become Ohio State and MD Anderson, which largely remains the case today. John Bird tested rituximab monotherapy using a more intensive dosing schedule due to the modest activity of the agent. That was published in 2001. The same year, Susan O'Brien explored higher dosing schedules at MD Anderson. But unbeknownst to many outside of MD Anderson, between November 1999 and December 2001, they piloted their first experience of the combination of fludarabine, cyclophosphamide, and rituximab called FCR. They would publish their initial findings in 2005, establishing FCR as the standard regimen for the next decade. Interestingly, there was no mention of molecular subgroups, FISH, or IGHV. That was not yet known when the study had started. Progression-free survival outcome was reported by response. Unfortunately, the median age in the study was 57 years old, and when this was exported to the community setting, where the median age at first therapy was 74, the toxicity was prohibitive. FCR really became a wedge between academic medicine, where it was embraced, and the community, where it was shunned. It was at the same time in England where Terry Hamblin identified the IGHV mutated versus unmutated groups envisioned decades earlier by David Galton and published in Blood in 1999. This figure shows the median survival of the disease from diagnosis was between 8 to 14 years, depending upon the IGHV status. The following year, Donor from the German CLL group introduced FISH testing, published December 2000 in New England Journal. Here, the median survival from diagnosis for high-risk patients was under 3 years to up to about 10 years for favorable FISH subgroups. Once again, it was the German CLL study group to unambiguously define a standard of care. The CLL-8 study was the first CLL study to ever demonstrate an overall survival benefit from therapy when they compared FC to FCR. But FCR was simply too toxic to be routinely given to typical CLL patients above age 70, and there was a need for less intensive therapy. Next development in the contemporary understanding of CLL had actually started decades earlier, but escaped the attention of Western investigators. Genopharm had been established in 1966 in East Germany when Germany was split after World War II. Virtually all of the pharmaceutical companies were in the West. The company was set up with scientific leadership, but primarily staffed by individuals in East Germany that were simply assigned to the company, not necessarily with scientific training. Initially, they produced antibiotics and they moved to steroid synthesis. And interestingly, using hog bile precursors, they developed a steroid doping program that was subject to decades of Olympic controversy. Eventually, they moved to chemotherapy as well, and upon hearing the results in the West, scientists at the Institute for Microbial Experimental Therapy produced bendamustine, envisioning a combination of an alkylating agent and a purine antimetabolite fused into a single molecule. IMET 3393 would be acquired by Genopharm and marketed as cytostasin. But bendamustine was never subjected to rigorous clinical development. It entered the market where it was used to treat Hodgkin's disease, breast cancer, myeloma, and other disease, and it essentially remained unknown to Western clinicians until after the fall of the Berlin Wall in 1989. The winding path of bendamustine is really remarkable. Over a course of several decades, it was owned by nearly a dozen different pharmaceutical companies, changing names along the way. As German clinicians began to perceive the potential value of the drug, Bruce Chesson received a phone call while sitting at his desk from a woman with a thick German accent asking him to come speak at a lymphoma meeting in Berlin. Having never been, Bruce decided to go, where he was presented with data from bendamustine. Fearing that the data was too good to be true, they hoped they could arrange a collaboration with a U.S. partner. The following year, Bruce convened a symposia in Portugal where all the available data on bendamustine could be reviewed. With the data collected, Bruce introduced the molecule to a San Diego-based company, Cellmetics, who licensed the drug and began US-based testing. As community physicians revolted against FCR, the introduction of bendamustine rituximab rapidly transformed the therapeutic landscape. The tolerability of the regimen was vastly superior to FCR, and a debate ensued, one that could only be answered by the Germans. The CLL-10 study was described by Susan O'Brien as the study where, quote, everybody wins. FCR was more effective, but also more toxic. Bendamustine rituximab had lower PFS, but was more tolerable. The PFS toxicity was a wash, and overall survival was the same. Bendamustine with rituximab quickly established itself as the most commonly used regimen in the United States. But rituximab was considered less active in CLL than other lymphoid malignancies, and there were efforts to improve the efficacy. In that context, Pablo Umaña set out to build a better mousetrap. Scientists had learned that the sugar molecules on the tail of an antibody known as glycosylation could impact the efficacy. Pablo was actually from Costa Rica and had obtained an undergraduate degree in bioengineering. He secured a position at Caltech where he started his PhD developing Chinese hamster ovary cells, commonly referred to as CHO cells, that can be modified to produce antibodies with varying levels of sugar attached. When his PhD advisor moved to Switzerland, Pablo moved with him. He unsuccessfully tried to sell the patent for the technology as biotech companies were skeptical, and one of his colleagues suggested he should start a company of his own, and that was Glycart. Some of the early investors pushed Umaña to develop their own antibody, so he set out to identify a validated target and make it better. He settled on CD20, changed the sugar molecules, identified it as a type 2 antibody as opposed to type 1, and the molecule obinutuzumab was born. One of his former board members had joined Roche and called him out of the blue, asking to acquire the molecule. Glycart was sold to Roche and clinical trials started. The first study enrolled from January 2008 to 2009 and was published in May of 2012. Roche presented the early data to Michael Halleck, and based upon fewer than 10 CLL patients, Michael pushed them to launch a phase 3 study, which enrolled from April 2010 to 2012. Remarkably, the final update of that study, an overall survival benefit of obinutuzumab over rituximab was identified. Obinutuzumab was approved November 2013 as the first agent with breakthrough therapy designation by the FDA. For a brief window of time, there was a balance of options based upon patient fitness: FCR for the young and fit, bendamustine rituximab for those in the middle, and obinutuzumab chlorambucil for the least fit. Michael Halleck commonly referred to the phrase go-go, slow-go, and no-go to describe such patients. Several months later, though, the CLL world would be turned upside down. I'd entered undergraduate at UC San Diego, vowing not to go to medical school, although I had majored in cellular biology and biochemistry. 3 courses, however, changed the direction of my life. I took courses in virology, immunology, and molecular basis of disease and decided that I would go to medical school. I joined a research laboratory at UC San Diego, which had cloned and identified the first tyrosine kinases. And even before entering medical school, I wondered if cancer might be treatable by interfering with the kinases. I ended up at UC Davis for medical school, and in 2000, I was doing an ICU sub-internship in my 4th year, and I took care of a young Ukrainian woman dying of CML. As she struggled on the ventilator with invasive sinus mucormycosis, undergoing facial surgeries that left her terribly disfigured, her case made a profound impression on me. I went to Mass General for residency, where I saw a few patients receiving a drug then known as STI 571 for chronic myelogenous leukemia. This was the original Gleevec study led by Brian Drucker, and Gleevec would represent a watershed moment in cancer as it revealed the power of targeted kinase inhibitors. When I arrived in Stanford in the fall of 2004, I was hungry for a research project. Sandra Horning had offered me a retrospective chart review on post-transplant lymphoproliferative disorder, and Ranjana Advani had asked me to undertake a project on NK T-cell lymphoma, 2 diseases linked together by Epstein-Barr virus. Recognizing the link between virology, hematologic malignancy, and aberrant kinases, I really dove into EBV biology, looking for ways that the virus could elucidate important biology. When EBV takes over cells, it does so with a limited repertoire of genes. And one that really stuck out to me was a protein called LMP2A. LMP2A sort of functionally mimicked the B cell receptor, and I realized that EBV was indeed pointing us towards a critical opportunity in B cell biology. I came to believe that if we could inhibit B cell receptor signaling, it might block malignant B cell growth. Right about then, a remarkable paper was published in which scientists showed that eliminating a portion of the B cell receptor would cause the mice to lose their B cells. In essence, if you block signaling from the B cell receptor, the cells died. In Ron Levy's lab, a postdoc named Jonathan Irish had utilized a new technique and showed that cancer cells actually had enhanced signaling through the B cell receptor, and in particular through a enzyme called SYK, SYK. I cold called a company called Rigel Pharmaceuticals making a SYK inhibitor and tried to persuade them that their drug could be used to treat lymphoma. While they thought the idea was interesting, they were developing a nasal spray version to treat allergies and an oral version for the treatment of rheumatoid arthritis. They agreed to provide me with a sample of R406, and I set out to work, presenting abstracts at the 2007 ASCO and ASH meetings, arguing in favor of B cell receptor signal inhibition. Based upon the preclinical data generated in our lab and similar efforts that started in Boston and Germany around the same time, we launched a trial of the molecule called fostamatinib in lymphoma. This would be presented in the plenary session of ASH in San Francisco at 2010. And this was the first place where we saw the shift in white blood cell count caused by B cell receptor signal inhibitors. As I treated patients with fostamatinib at Stanford, one of my attending physicians, Richard Miller, who had co-founded Biotherapy Systems with Ron Levy, saw my patients and was impressed. I explained to him why B cell receptors should work, and he had actually picked up a compound from another company that was called PCI 32765, which they weren't really planning at that time to turn into a drug. But his company called Pharmacyclics was nearly bankrupt, and he decided to bet the farm on his BTK inhibitor. So as a fellow, we set up a consulting agreement, and I helped design the first in human phase 1 study. I left for a community practice job, and the project was given to a fellow in the class behind me, but the study was very slow to enroll, and Pharmacyclics asked if I would agree to open it within U.S. Oncology. The very first CLL patient treated was in my small clinic in Springfield, Oregon, and the protocol was written to take the medication 4 weeks on, followed by 1 week off. This resulted in almost like a sawtooth pattern of lymphocytes going up and then falling down. The lymphocytosis concerned the medical monitor, so only a few days after starting the medication in patient 1, we got a CT scan that showed a nice reduction in the lymph nodes. The on-off impact of lymphocytosis honestly made it difficult to understand if we were even helping patients initially. Nodes and blood counts would yo-yo depending on whether they were taking the drug or having an off week. So we amended the protocol and gave continuous dosing. And this is the graph from a patient of one of my colleagues who transitioned from intermittent to continuous dosing therapy. The first in human study was published in the Journal of Clinical Oncology in 2013 with 3 authors from our community practice network. As we developed the early data, I was asked to present our findings to a group of key opinion leaders gathered in Palo Alto. John Bird, Susan O'Brien, Steve Coutre came to hear about the early experience of BTK inhibitors in CLL, and we designed the next study. Unfortunately for me, the program was embraced by so many academic centers that our participation was virtually irrelevant to the future clinical development. Based upon the phase 2 data for ibrutinib, the drug was approved in relapse and refractory disease, but it would be first of nearly a dozen BTK inhibitor studies published in the New England Journal. Recognizing that this project had grown far larger than myself, I had to pivot, and John Bird helped me get involved with idelalisib, and our group ended up leading those registration studies. 3 months after the approval of obinutuzumab, ibrutinib was approved in chronic lymphocytic leukemia. The Business of BTK Inhibitor Development was recently published in a top-selling novel, For Blood and Money by Nathan Vardy. A second book was published in summer of 2024 entitled Race for a Remedy that addressed the scientific early days of BTK inhibitors. But several landmark studies followed quickly thereafter. BTK versus ofatumumab in July 2014 led to the full approval in relapse setting, and BTK beat chlorambucil in December 2015, leading to the full approval in the frontline setting. Of course, beating up on ofatumumab in relapse disease or chlorambucil in the treatment-naive patients expanded the label, but there was controversy regarding the optimal frontline treatment with the efficacy of more aggressive cytotoxic regimens such as bendamustine or FCR. ASH of 2018 was a watershed year in CLL. However, as 2 frontline studies read out in the plenary session and late-breaking abstract session, BTK beat FCR and BTK beat bendamustine rituximab. Both were simultaneously published in the New England Journal. Second-generation BTK inhibitors acalabrutinib and zanubrutinib would then directly compete against ibrutinib, showing improved safety and/or efficacy in relapse disease. I had the opportunity to lead the frontline study of acalabrutinib, leading to full approval of this agent, and zanubrutinib would be BR gaining frontline approval. Unfortunately for me, I have to skip over the PI3 kinase inhibitor story, but they play only a bit part in CLL management today. But BCL2 would be the next critical therapy developed. A human produces approximately 60 billion cells per day and an equal number of die. Apoptosis is actually a Greek term to define leaves falling in autumn. It was used 2,000 years ago by Hippocrates to describe tissue death as scabs fell from wounds. Virchow observed cell death and used the term in his writings, but it was first used by modern biologists in 1972 as initial observations of programmed cell death were made in Australia. Little was written about apoptosis until 1984 when BCL2 was identified in Carlo Croce's lab and found to protect cells from death. Other BCL2 family members were identified, and a 1995 Blood article identified BCL-XL as one of the most important markers of chemotherapy resistance. At the time, in 1996, the field was dominated by chemotherapy, but research scientists at Abbott Laboratories were interested to see if they could develop a drug to inhibit the BCL2 and BCL-XL proteins. This began a 20-year project inside the company involving an entire team of chemists, biologists, toxicologists, and more to create what we now know as venetoclax. Saul Rosenberg would lead the project from ABT 737 through navitoclax, and Andrew Sowers would lead from navitoclax to ABT 199, which is what we now know as venetoclax. Their initial focus was on BCL-XL for the chemotherapy resistance. First, they had to create a crystal structure, and the following year they did the same when it was precipitated with another protein that was important for the biology. They identified a key groove where the two proteins interacted and set out to create a drug that would do the same thing. This was really pioneering work, as no drug had ever been developed specifically to block the interface of two separate proteins. At the time, creating enzyme inhibitors were much more common, but targeting protein-protein interactions was rarely done. Rather than a deep binding pocket, the more superficial, complex-shaped surfaces of protein binding sites were not considered drug targets. They use a very elegant screen and found a way to eventually make a drug that inhibited both BCL2 and BCL-XL. But unfortunately, when they gave it in early clinical trials, patients' platelets suddenly dropped. And when they tried combining it with chemotherapy, the effect on blood counts was simply too toxic. The data would be presented in ASH 2008 and then published in the JCO in 2011. And here you can see the marked reduction in platelets here, and caused by the agent, but you can also see the impressive reduction of the CLL cells. So the team realized they needed to pivot and re-engineer the molecule to dial out the BCL-XL and dial up the BCL2. During an afternoon conference session, one of the scientists stared at the crystal structure and proposed moving this portion of the molecule over to here and swapping it out. And patients treated in the phase 1 with a starting dose of 100 milligrams. In the initial publication, they reported 3 patients treated with profound drops in lymphocyte counts, but also the emergence of tumor lysis syndrome. Today, we start with 20 milligrams and increase. Once again, the German group led key pivotal studies. The Murano study, published in the New England Journal, compared venetoclax rituximab to BR in relapsed refractory CLL, leading to approval. The German group also led CLL-14 of obinutuzumab venetoclax, which was compared to obinutuzumab chlorambucil, leading to the approval in the frontline setting. The CLL-13 study was recently presented, which compared venetoclax with either rituximab or obinutuzumab to aggressive chemotherapy regimens such as FCR or BR. Once again, obinutuzumab was shown to be superior to rituximab, and the combination of obinutuzumab's venetoclax was superior to chemotherapy. Naturally, interest in BTK and BCL2 inhibitor combinations would arise. MRD-guided therapy with ibrutinib venetoclax was compared to FCR, demonstrating clear superiority of ibrutinib venetoclax compared to obinutuzumab chlorambucil, and the combination was approved in many areas, but not in the United States. The 2 most important studies ongoing today to define the optimal frontline study include the MAGIC study, which compares acalabrutinib venetoclax to obinutuzumab venetoclax with the primary endpoints of MRD and PFS. This will really help define the optimal doublet therapy for fixed duration treatment. The second study is the German CLL-17 study, which will look at indefinite ibrutinib versus fixed duration obinutuzumab venetoclax or ibrutinib venetoclax. At ASH 2024, we saw the results of acalaven with or without obinutuzumab, and we're anticipating that that will be approved in the US fairly soon. Beyond these agents, however, there are novel strategies emerging. We've seen the recent approval of pirtobrutinib, a third-generation BTK inhibitor, which does not utilize covalent inhibition, but has superior pharmacokinetics, enabling activity in the setting of common BTK resistance mutations. In addition, fourth-generation BTK inhibitors, which cause BTK degradation, even in the setting of additional mutations, looks like the next exciting evolution in BTK targeting drugs. Furthermore, we're on the edge of scientific advances in immunotherapy. CAR T has been approved in multiple hematologic malignancies and was approved in CLL within the last year. I now have several patients treated with CAR T, and I think we may be on the edge of seeing some patients actually cured of their disease. Off the shelf bispecific antibodies have been approved in multiple conditions, and we're now making legitimate headway in CLL as well. So CLL was discovered by Virchow at the end of the 19th century, and Ehrlich created modern hematology shortly thereafter. George Minot described the first series of cases approximately 100 years ago, and for almost 50 years, treatment was radiotherapy. From mustard gas to the synthesis of chlorambucil in the 1950s, we really didn't have anything more effective for yet another 40 years. Fludarabine almost died in phase one due to a dosing miscalculation, but became the platform for combination chemotherapy with FC, then chemoimmunotherapy with FCR. Bendamustine was, quote, discovered, end quote, after the Cold War and quickly became the US standard. Obinutuzumab, the vision of a Costa Rican bioengineer transplanted to Switzerland, would displace rituximab, and we've really only had access to BTK inhibitors for 10 years and BCL2 inhibitors for less. We now have second and third generation BTK inhibitors that have only just recently been approved along with CAR T. So has the mission been accomplished yet? While we've made extraordinary and accelerating progress, much of which has been in only the last 15 years, patients want to be cured. I believe that we have the tools in hand now to get there. And while work remains to be done, we're getting very close. Thank you for your time.
That was fantastic, and you did such a good job getting through that. I'm so
It's a
impressed.
lot of material. I'm sorry, but
Yeah, no,
uh,
that was fascinating. I didn't know honestly the majority of that sort of history, and studied I have a research background from the University of Utah where I spent a lot of time in the Winthrop building. Like
there you go.
I've been around the history of hematology, and all of that was so new to me. So truly that was fascinating. Um, I don't want to hold you too long, so I don't know if we'll have time for questions,
Yeah, I'll
but
probably have
just
to I'll have to hold it there because I actually do have another meeting here. Part of why I was going
um,
so fast, but I
oh,
do thank you for the time to present this, and thank you for coordinating this.
absolutely. And I'll just say that if you let me know when your book is published, I would be happy to make sure that our audience is aware of that because I speak for myself and I assume I speak for others here when I would love to read that. And I think everyone else would as well. So if you keep me posted on when that's published, I'll make sure that the word gets out.
Super. Thank you for your time, Dalton.
Absolutely.
Bye bye.
All right. Bye. All right, and we're going to take just a couple moments here to wrap up. Um, so I wanted to highlight the Track My Disease feature. Actually, am I sharing? I am sharing. I wanted to highlight the Track My Disease feature in the HealthTree Care Hub. So once you've connected your medical records, you can use the Track My Disease feature to provide you with the ability to gain a one-of-a-kind access to your health information. This will encompass your lab results, genetic profile, and treatment histories, offering you invaluable insights into your own medical journey. On the screen, I have the link to sign up for the HealthTree Care Hub, along with the email address of our amazing patient navigators who are able to create an account and connect to your records. I'll also include information about how to enroll in the HealthTree Care Hub in our follow-up email, so be on the lookout for that. And then we do have quite a few upcoming events for the month of April. Actually, this morning, I recorded a podcast with Dr. Joanna Rhodes. This is about BTK degraders, which Dr. Sharman mentioned towards the end of his talk there. So if you are interested in learning more about that and kind of this new wave of therapies that's being developed, I have an upcoming podcast about that. And then we have a roundtable that'll be on April 7th at noon Eastern. This is Understanding Chronic Lymphocytic Leukemia Treatment Sequencing. This will have three different doctors, Doctors Nicole Lamana, Bita Fakhri, and Dr. Debbie Stevens. And then another webinar will be happening on April 16th. And this one will be talking about cardiotoxicity and BTK inhibitors, what CLL patients should know. And that will be with Dr. Carrie Rogers from Ohio State. So Thank you again to our sponsor, AbbVie. Truly, we would not be able to host these types of events without them. So, big shout out to AbbVie. And finally, I just want to say thank you all to helping us build our CLL community. I've gone ahead and added my email to this slide. Don't hesitate to reach out to me if you have any questions about HealthTree or any of our programs, or even if you just want to chat. I'm always available for you. And then I appreciate you all, and I hope you have a great rest of your day. Hope to see you at another webinar soon. Bye.